Hello everyone. Today we are discussing about the fourth chapter, the importance of high energy bond, in the part 1 of the textbook Molecular Biology of the Gene by Watson et al. Each time a potential weak bond was considered, a question was arised. Does its formation involve a gain or loss of free energy? Only when delta G is negative does the thermodynamic equilibrium favor a reaction. This same approach is equally valid for covalent bonds also. The fact that enzymes are usually involved in the making and breaking of a covalent bond does not in any way or in any sense alter the requirement of a negative delta G. So first we are discussing about general thermodynamic properties of covalent bonds. There is great variation in the amount of free energy possessed by specific molecules because covalent bonds do not all have the same bond energy. For example, the covalent bond between oxygen and hydrogen is considerably stronger than the bond between hydrogen and hydrogen or oxygen and oxygen. The formation of an OH bond at the expense of OO, O bond O, or H bond H will thus release energy. So, sufficiently concentrated mixture of oxygen and hydrogen will be transformed into water. Thus, a molecule possesses a large amount of free energy if linked together by weak covalent bonds than if it is linked together by strong bonds. So the molecules which possess large amount of free energy if they are linked together by weak bonds. So the molecules which are linked together by weak bonds are having large amount of free energy than the molecules which are linked together by strong bonds. This idea at first glance gives the notion that the stronger the bond, the less energy it can gives off. But it can be realized that an atom that has formed a very strong bond has already lost a large amount of free energy in this process. So, in order to form a strong bond, that molecules which are involved in the formation of that strong bond has already given out or already lost a large amount of free energy in that process. Therefore, the best food molecules, that is, the molecules that donate energy, are those molecules that contain weak covalent bonds and are therefore thermodynamically unstable. For example, glucose is an excellent food molecule because there is a great decrease in free energy when it is oxidized by oxygen to yield carbon dioxide and water. On the other hand, carbon dioxide composed of strong covalent double bonds between carbon and oxygen known as carbonyl bond is not a food molecule in animals. In the absence of energy donor ATP, carbon dioxide cannot be transformed spontaneously into more complex organic molecules, even with the help of specific enzymes. Carbon dioxide can be used as a primary source of carbon in plants only because the energy supplied by light quanta during photosynthesis results in the formation of ATP. The chemical reaction by which molecules are transformed into other molecules containing less free energy do not occur at significant rate at physiological temperature in the absence of catalyst. This is because even a weak covalent bond is, in reality, very strong and is only rarely broken by thermal motion within a cell. For a covalent bond to be broken, in the absence of a catalyst, energy must be supplied to push apart the bonded atom. When the atoms are partially apart, they can recombine with new partners to form stronger bonds. In this process of recombination, the energy released is the sum of free energy supplied to break the old bond plus the difference in free energy between the old and new bond. Here in this picture, they try to explain the energy activation of chemical reactions. Here we can see that these two reactors, A bond B, C bond D, that is A, B and C, D are the reactants that undergo reaction to form the products A, D and C, D. And in order to convert these reactants into products, these reactants should reach or that has to reach a particular state called as activated state with the expense of activation energy, and after that the products will be formed. And when this reactants converted to products, the free energy will be released. Okay, that will be always the free energy will be decreased. Delta G is always negative. The energy that must be supplied to break the old covalent bond in a molecular transformation is called as the activation energy. So the energy which is required or that has to be supplied to break the old covalent bond, the energy that has to be supplied to break the old covalent bond, the covalent bond between A and B and C and D is broken with the help of or by utilizing the energy that has been provided into the system, and that energy is called as activation energy. It is usually less than the energy of original bond because molecular rearrangements generally do not involve the production of completely free atoms. Instead, a collision between the two reacting molecules is required, followed by the temporary formation of a molecular complex called as activated state. In the state, the close proximity of the two molecules makes each other's bond more labile, so that less energy is needed to break a bond than when the bond is present in free molecules. Okay, first of all, these reactants will reach into a state called as activated state. At that time, the bond which are involved in keeping these molecules together will be more labile, and that will be easily broken apart for the formation of products like AD and CD. Most reaction of covalent bonds in cells are therefore described as A bond B plus C bond D gives A bond D plus C bond B. And the mass action expression of such reaction is always KEQ is equal to product of concentration of products divided by product of concentration of reactants. That means KEQ is equal to concentration of AD into concentration of CB divided by concentration of AB into concentration of CD. We know that KEQ is the equilibrium constant, and the KEQ is always related to delta G by delta G is equal to minus RT log KEQ, or KEQ is equal to E raised to minus delta G by RT. That we have already discussed in the previous video, so I'm not going into the details. Okay. Then, because of energies of activation are generally between 20 and 30 kilocalorie per mole, activated states practically never occur at physiological temperature. Okay, so since the activation energy of most of these molecules are in between 20 and 30 kilocalories per mole, so the activated states of the molecules are practically never occur at physiological temperature. High activation energy are thus the barriers preventing spontaneous rearrangement of cellular covalent bonds. Next, we will look into the details of how enzymes lower this activation energy in biochemical reactions. The function of enzyme is to speed up the rate of a chemical reaction by lowering the activation energies of molecular rearrangements to values that can be supplied by the heat of motion. When specific enzyme is present, it can cause a rapid formation of the reactants possessing the lower amount of free energy. So, enzymes can never affect nature of an equilibrium. They merely speed up the rate at which it is reached. Thus, if thermodynamic equilibrium is unfavorable for the formation of a molecule, the presence of an enzyme cannot bring about the reaction. And this idea can be explained based on this picture. And in this picture, they have explained enzymes lower activation energies and the speed up the rate of reaction. Here, the purple colored diagram or the purple colored curve denotes the involvement of enzymes to reduce the activation energy. So, this is the curve that represents the reaction under catalytic activity of enzyme, and this red colored one is the uncatalyzed reaction. Activated energy is required for this reactants to get converted into products, and this in the absence of this catalyst or the enzyme. But this activation energy of this reactants can be reduced to this stage with the involvement of this catalyst or the enzyme. But here we can see that this delta G remains the same or the enzymes do not have a role to play in decreasing the or altering this delta G. Okay, so note that delta G remains the same because the equilibrium position remains unaltered. So, if the thermodynamic equilibrium is unfavorable for the reaction to or the for the formation of a molecule, the presence of an enzyme cannot bring about the reaction at all. Then comes the free energy in biomolecules. Thermodynamics tells us that all biochemical pathways must be characterized by a decrease in free energy. This is clear in the case of degradative pathways, in which thermodynamically unstable food molecules are converted to more stable compounds, such as carbon dioxide and water, with the evolution of heat. All degradative pathways have two primary purposes. First purpose is to produce small organic fragments necessary as building blocks for larger organic molecules. Second one is to conserve a significant fraction of the free energy of original food molecule in a form that can do work, which is accomplished by coupling some of the steps of degradative pathway with the simultaneous formation of high energy molecules such as ATP, which can store free energy. So the two primary purposes of all degradative pathways are: first one, to produce small organic fragments which are functioning as building blocks for larger organic molecules form. And the second one is to convert significant fraction of this free energy into storage form of energy, that is nothing but the ATP. Not all free energy of a food molecule is converted into free energy of high energy molecules. All degradative pathways are characterized by a conversion of at least 1/2 the free energy of the food molecule into heat and or entropy. For example, it is estimated that in cells, approximately 40% of the free energy of glucose is used to make new high energy compounds, the remainder being dissipated into heat energy and entropy. So, next we are discussing about high energy bonds hydrolyzes with large negative delta G. The breakdown of a high energy molecule containing one or more bonds by water is called hydrolysis. Hydrolysis is usually accompanied by a large decrease in free energy. The specific bonds whose hydrolysis yield these large negative delta G are called as high energy bonds. These are here one thing to remember that it is not the bond energy but the free energy of hydrolysis is high, and it is represented using the symbol tilde. This symbol is tilde, and it is also called as Siegel. So the high energy bonds are usually expressed using tilde. The energy of hydrolysis of average high energy bond is 7 kilocalorie per mole. The most important high energy compound is ATP, and it is formed from inorganic phosphate and ADP using energy obtained from degradative reaction or from sun. Many other high energy compounds are there. There are lots of different forms of high energy compounds are there. Some are directly formed during degradative reaction. Others are formed during or using some of the free energies of ATP. And this table shows some important classes of high energy bonds and their delta G values of change in free energy during its reaction. First class is pyrophosphate. And here pyrophosphate is nothing but two phosphate molecules are connected with the help of a high energy bond. Okay, and that the pyrophosphate undergo reaction to release two phosphate groups. And the delta G value of that reaction will be minus 6 kilocalorie per mole. Next class is nucleoside diphosphate. Example is adenosine diphosphate, adenosine plus 2 phosphate groups. And adenosine diphosphate undergo reaction to form AMP plus phosphate. With the delta G value minus 6 kilocalorie per mole. Nucleoside triphosphate, adenosine plus 3 phosphate, that is ATP. ATP undergo reaction to form ADP plus phosphate, and the ATP can also converted to AMP plus phosphate, AMP plus pyrophosphate, AMP plus pyrophosphate. So by the splitting of this ATP, the delta G value will be minus 7 kilocalorie per mole. Enol phosphate. Example is phosphoenol pyruvate. The phosphoenol pyruvate will be converted to pyruvate plus phosphate with the free energy of minus 12 kilocalorie per mole. Next class is amino acyl adenylate. And this amino acyl adenylate, that is amino acid plus this AMP. Amino acid plus AMP. Okay, so amino acid plus AMP. And that undergo hydrolysis or reaction to form AMP plus amino acid with the release of free energy minus 7 kilocalorie per mole. Vanadium phosphate. Example is creatine phosphate. Creatine plus 2 pyrophosphate, that will give of creatine plus phosphate. The delta G value of minus 8 kilocalorie per mole. Thioesters. Example is acetyl coenzyme A. Acetyl coenzyme A will be converted to coenzyme A plus acetate with the free energy change of minus 8 kilocalorie per mole. Most of the important types of high energy bonds involve either phosphate or sulfur. From this entire class of high energy bonds, we could see that most of these classes are having phosphate or sulfur. Phosphate or sulfur as compound that either consist of phosphate or sulfur atoms. The high energy pyrophosphate bonds of ATP arise from the union of phosphate groups. Pyrophosphate, as we have already seen, pyrophosphate is an association of two phosphate groups. So it is formed by the association or union of two phosphate groups. In another high energy phosphate bond, phosphate group attached to oxygen atom of a carboxyl group create high energy acyl bond. Here, in this picture, here the phosphate group of this adenosine or adenosine monophosphate is attached to the oxygen of COO or carboxyl group of a compound. A compound. So that type of bond is called as acyl bond. This acyl bond is also considered as a high energy bond. High energy bond involving sulfur atom plays an important role, just like that of phosphate, in energy metabolism. The most important molecule containing a high energy sulfur bond is acetyl coenzyme A. So acetyl coenzyme A is one of the best example for a sulfur containing high energy bond. This bond is the main source of energy for fatty acid biosynthesis. The high energy bonds show wide range of delta G values. Okay, here from this description itself, we can see that the delta G values of this high energy bond is highly variable. So the criteria that calling a bond high energy is whether its hydrolysis can be coupled with another reaction to affect an important biosynthesis. For example, the negative delta G accompanying the hydrolysis of glucose 6-phosphate is 3 to 4 kilocalorie per mole. But this delta G is not sufficient for efficient synthesis of peptide bonds. So this phosphate ester bond is not included among high energy bonds. Okay. Then high energy bonds in biosynthetic reactions. Many biosynthetic pathways demand an external source of free energy. The free energy sources are the high energy compounds. The making of many biosynthetic bonds is coupled with the breakdown of high energy bonds, so that the net charge of free energy is always negative. Thus, high energy bond itself generally have a short life. Almost as soon as they are formed during a degradative reaction, they are enzymatically broken down to yield the energy needed to drive another reaction to completion. And one more thing is that not all steps in biosynthetic pathways require the breakdown of a high energy bond. Often, only one or two steps involve such a bond. In the remaining step, delta G is effectively zero or may even be slightly positive. These small positive delta G values, however, are not significant, so long as they are followed by a reaction characterized by the hydrolysis of a high energy bond. Rather, it is a sum of all the free energy changes in a pathway that is significant. It does not really matter that the KEQ of a specific biosynthetic pathway is slightly in favor of degradation if the KEQ of the succeeding step is in favor of the forward biosynthetic direction. Okay, and this concept can be better explained with this example, this picture. Here in this picture, they have shown that the free energy changes in the multi-step metabolic pathway. That means the substrate A is converted to an intermediate B, and then B again converted to an intermediate C, C again converted into an intermediate D, and the D ultimately converted into the product E. Okay, two steps. Here you can see the two steps, that is A to B and C to D. Here in this A to B and C to D, that will do not favor the A to E direction of the reaction because they have small positive delta G value. And the conversion step of A to B and C to D There we can see there is a delta G change in a positive direction, slight positive direction. So these two steps will not favor the conversion of A into E. However, they are insignificant owing to the very large negative delta G value provided in steps B and C, B to C and D to E. And these positive delta G values during the conversion of A to B and C to D are insignificant because the very large negative delta G values during the conversion of B into C and D into E. So that will nullify the positive delta G values during the conversion of A to B and C to D. Therefore, the overall reaction favor the A to E conversion. In short, it does not really matter that the KEQ of a specific biosynthetic step is slightly in favor of degradation. Okay, so here the delta G, positive delta G values, it does not matter. These two steps, even though these two steps are having slight positive delta G values, since there is high delta G negative values during the succeeding steps, the slight positive delta G values does not make any problem, or if they are insignificant. Likewise, not all the steps in degradative pathways generate high energy bonds. For example, only two steps in glycolytic, that is Embden-Meyerhof pathway, breakdown of glucose generate ATP. So only two steps in the glycolytic pathway, the ATPs are generated. It uses up 2 molecules of ATP for every 4 that is generated. Okay, so in short, 2 ATPs will be produced as an after effect of glycolytic pathway because 2 ATPs have been already consumed during this pathway. As in every energy yielding degradative process, more high energy bonds must be made than consumed. So this is common to all degradative pathways or energy yielding degradative pathway because more high energy bonds must be made than consumed. Then coming to the details of peptide bond hydrolysis. Peptide bond hydrolysis. The formation of a dipeptide and a water molecule from 2 amino acids require a delta G of 1 to 4 kilocalorie per mole depending on which amino acids are being joined. These positive delta G values by themselves tells us that the peptide chain cannot form from 3 amino acids. Okay. Here we know that the dipeptides are usually formed by the association of 2 amino acids. Amino acid A and amino acid B form dipeptide, peptide bond to form AB, that is a dipeptide. Here, the delta G values during this reaction will be in the range of 1 to 4 kilocalorie per mole. That are positive, they are having positive delta G values. Just now we have seen that in order to carry out a reaction, there should be the delta G values should be negative. Okay, decrease in delta G value should happen or should be negative. So in this from this positive value of delta G during the formation of a dipeptide or a polypeptide chain, we can simply say that that will say that the peptide chain cannot form from 3 amino acids. The energy must be added to amino acids before they can be united to form protein. So the energy should be added to overcome this positive delta G value in order to form the polypeptide chain. It is usually done by ATP. And the ATP contains 3 phosphate groups. ATPs usually contain 3 phosphate groups attached to an adenosine molecule. The ATP contains 3 phosphate groups attached to adenosine. When 1 or 2 of the terminal phosphate groups are broken off by hydrolysis, there is a significant decrease in free energy. When 1 or 2 of this terminal phosphate of this ATP has been hydrolyzed or removed or broken off by hydrolysis, their yield or their causes severe decrease in free energy. So from this equations, we can see that adenosine triphosphate upon hydrolysis yield adenosine diphosphate plus phosphate. At that time, the change in free energy will be, or the delta G value will be minus 7 kilocalorie per mole. When this adenosine triphosphate undergo hydrolysis to yield adenosine monophosphate plus pyrophosphate, at that time the delta G value will be minus 8 kilocalorie per mole. When this adenosine diphosphate undergo hydrolysis, it will yield adenosine monophosphate plus a phosphate group, and with the delta G value minus 6 kilocalorie per mole. All these breakdown reactions have negative delta G value considerably greater in absolute value than the positive delta G value, accompanying the formation of polymeric molecules from their monomeric building blocks. And all these hydrolysis reaction of this high energy compound, that is ATP, we got the decrease in free energy, or the negative delta G values, which are considerably greater in absolute value than the positive delta G values accompanying most of the polymeric molecules. Okay. Just now we have seen that the formation of a polypeptide or a dipeptide, the delta G values are usually in between 1 to 4, that is positive 1 to 4. And from this picture itself, and we have one more thing we have mentioned, that there should be the ATP, or the energy should be, or free energy should be supplied to this amino acid in order to carry out the process of peptide linkage or the formation of peptide linkage. Okay, so from this details we can see that here the delta G values are 7, 8. Absolute values of this delta G are sufficiently enough or sufficiently large enough from this positive values of of most of this positive values during the formation of polymeric molecules from their monomeric building blocks. The essential trick underlying these biosynthetic reactions, which themselves have a positive delta G, is that they are coupled with the breakage of high energy bond, characterized by negative delta G of greater absolute value. So the essential trick underlying all these biosynthetic reactions, consider the case of the formation of a polypeptide chain, and during the delta G value for the during the formation of dipeptide is usually 1 to 4, that is positive, and this will be overcome by the free energy that are released during sufficiently large free energy that are released during the hydrolysis of ATP. Okay, so by coupling these two reactions, that means the biosynthetic reaction, that is peptide formation or the polypeptide formation, along with this ATP breakdown, by coupling these two reactions, the positive value of this the formation of polymeric molecules can be overcome. Okay. Thus, during the protein synthesis, the formation of each peptide, that is delta G is equal to 0.5 kilocalorie per mole, is coupled with a breakdown of ATP to AMP and pyrophosphate, which has a delta G value of minus 8 kilocalorie per mole. This results in net delta G of minus 7.5 kilocalorie per mole, more than sufficiently to ensure that the equilibrium favors protein synthesis rather than breakdown. Okay. So during protein synthesis, the formation of each peptide bond is having a delta G value of plus 0.5 kilocalorie per mole. Okay, so from that positive value, we can see that it is very difficult to form the polypeptide or a peptide bond without the investment of free energy from somewhere. And we have already seen that that is usually coupled with conversion or the breakdown of ATP to AMP and pyrophosphate. We know that by the hydrolysis of ATP to AMP and pyrophosphate, the delta G value is minus 8 kilocalorie per mole. So in short, the net delta G value will be minus 7.5 kilocalorie per mole. That is more than enough for the formation of a peptide. So the reaction will favor the formation of polypeptide chain rather than the breakdown. Next comes the activation of precursor in group transfer reaction. When ATP is hydrolyzed to ADP and phosphate, most of the free energy is liberated as heat, which cannot be used to make covalent bonds. So, a coupled reaction is achieved by two or more successive reactions. These are always group transfers. The reactions not involving oxidation or reduction, in which molecules simply exchange the functional group. The enzymes that catalyzes this reaction are called as transferase. Here, we can see that these two molecules, A bond X, B bond Y, will be converted to A bond B, X bond Y. Here, the group X is exchanged with component B, and vice versa. The group Y is exchanged with component A. Okay, and the group transfer reactions are simply the exchange of functional groups, not any reduction or oxidation. And the group transfer reaction involving water is called as hydrolysis with the involvement of an enzyme hydrolysis. Here, the A bond B plus H bond OH. Here, the OH functional group of this compound or the molecule will be exchanged with A. So it forms AOH, and the H is exchanged with B. So it forms BH. So the group transfer reactions with the involvement of water with the help of this enzyme hydrolase is called as hydrolysis. In group transfer reactions involving groups attached to high energy bonds, the high energy group is transferred to an appropriate accepted molecule. So this appropriate accepted molecules becomes attached to the in group transfer reactions involving groups attached to high energy bonds, the high energy group is transferred to an appropriate acceptor molecule. It becomes attached to the acceptor by a high energy bond. So the group or the functional group is attached to this acceptor molecule with the help of a high energy bond. So group transfer allows the transfer of high energy bond from one molecule to another. So the group transfer involves the exchange or the transfer of a high energy bond from one molecule to another. For example, energy present in ATP transferred to form GTP, guanosine triphosphate, one of the precursors which is used in RNA. So one of the precursors, that is GTP, used in RNA synthesis is formed by the transfer of high energy or the energy containing group from adenosine triphosphate to guanosine. In this equation, they have shown that adenosine triphosphate plus guanosine monophosphate will yield adenosine, one of the terminal phosphate of this ATP will be given to this guanosine monophosphate. So the adenosine triphosphate will be converted to adenosine diphosphate, and the guanosine will be converted to guanosine diphosphate. The adenosine triphosphate plus guanosine, another molecule of adenosine triphosphate will be undergo group transfer reaction with guanosine diphosphate to form adenosine diphosphate plus guanosine triphosphate. So it can directly, this guanose and triphosphate can be directly used for the precursor for arabesque. The high energy pyrophosphate group. Pyrophosphate group is nothing but the PP. 2P. Pyrophosphate group on GTP allow it to unite with another molecule. So it is a high, since it is a high energy molecule, it can simply associate with another molecule. GTP is thus an example of an activated molecule. So the guanosine triphosphate GTP can be considered as a activated molecule. And the process of high energy group is called as group activation. And this process is called as group activation. Then comes the ATP versatility in group transfer reaction. In both oxidative and photosynthetic photophosphorylation, energy is used to synthesize ATP from ADP and phosphate. Here, adenosine triphosphate plus energy gives, here, in both oxidative and photosynthetic photophosphorylation, energy is used to synthesize ATP from ADP and phosphate. And the adenosine here we know that this is the adenosine diphosphate, and when a phosphate is added with the expense of energy, that will be converted to adenosine diphosphate will be converted to adenosine triphosphate. Because ATP is the original biological recipient of high energy group, it must be the starting point of a variety of reactions in which high energy groups are transferred to low energy molecules to give them potential to react spontaneously. Because the ATP is the original biological recipient of high energy group, it must be the starting point of a variety of reactions in which high energy groups are transferred to low energy molecules to give them the potential to react spontaneously. And as we know, the ATP contains two high energy bonds. Post-splitting releases three important specific groups like pyrophosphate. So the ATP contains two high energy groups. This is the first high energy bond. This is the second high energy bond. So when the breakage of this bond happens, what will be the result? The adenosine monophosphate, that is AMP. AMP plus pyrophosphate will be the result. And when this bond is broken, what will be the result? ADP. The ADP, adenosine diphosphate plus a phosphate is the result. So from this ATP, by the breakage of either of these two high energy bonds, we will get three important specific groups like pyrophosphate, adenosine monophosphate, or the phosphate. It is important to notice that these high energy groups retain their high energy quality only when transferred to an appropriate acceptor molecule. For example, the transfer of phosphate group to COO group yield a high energy COO phosphate, that is acyl phosphate group. The transfer of the same group to a sugar hydroxyl group, that is EOH, as in the formation of glucose 6-phosphate, that gives rise to a low energy bond, that is less than 5 kilocalorie per mole, decrease in delta G upon hydrolysis. So based on the acceptor molecule, the high energy groups retain this high energy quality based on the acceptor molecule. Okay, so as we have seen that the transfer of phosphate to a COO group yield a high energy acyl phosphate group, but the transfer of the same group to a sugar hydroxyl group, that is COH, as in the formation of glucose 6-phosphate, that will, okay, that will give rise to only a low energy bond, because upon hydrolysis of this glucose 6 phosphate, there will be a release of delta G less than 5 kilocalorie per mole. Next is the activation of amino acids by attachment of adenosine monophosphate. The activation of an amino acid is achieved by transfer of an adenosine monophosphate group from an ATP or an adenosine triphosphate to COO minus group, that is carboxyl group of this amino acid. In this picture, they have shown that this is an amino acid having a side chain R. This is the N terminal or the amino terminal, and this is the carboxy terminal. And adenosine monophosphate. An adenosine monophosphate from an adenosine triphosphate will be attached to the oxygen of the carboxyl group of this amino acid. At the same time, a pyrophosphate will be released. Okay, so the enzymes that catalyzes this type of reaction are called as amino acid synthase. So the activated amino acid will be formed. This activated amino acid undergoes a second group transfer at its carboxyl group to the end of a tRNA molecule to form amino acid tRNA. That is an activated form of amino acid. That means the amino acid that is attached with an adenosine monophosphate will be undergo the second round of group transfer with tRNA to form an amino acid tRNA molecule along with the release of adenosine monophosphate. A peptide bond then formed by the condensation of amino acid tRNA molecule onto the end of a growing polypeptide chain. So this amino acid tRNA will then added to a growing polypeptide chain with n number of amino acids to form a growing polypeptide chain with n plus 1 amino acid with the release of tRNA. The final step of this coupled reaction, like that of all other reactions, involves the removal of this activating group and the conversion of high energy bonds into one with a lower free energy of hydrogen. This is a source of negative delta G that drives the reaction in the direction of protein. Okay. Then we are going to discuss nucleic acid precursors are activated by the presence of pyrophosphate. Both the DNA and RNA are built up from mononucleotide monomers or nucleoside phosphates. The phosphodiester bonds that link nucleic acid release considerable free energy upon hydrolysis, approximately 6 kilocalorie per mole. The immediate precursor for both DNA and RNA are the nucleoside 5' triphosphates. For DNA, the precursors are DATP. D stands for deoxy, so it is deoxy adenosine triphosphate, DGTP, DCTP, and DGTP. For RNA, the precursors are ATP, GTP, CTP, and UTP. ATP serves as the main source of high energy groups in group transfer reaction and also as the direct precursor of RNA. So this ATP carries out two functions. It is the main source of high energy group in group transfer reaction and also the direct precursor of RNA. The other three RNA precursors, that is GTP, CTP, and UTP, arise by group transfer reaction, like that we have already discussed in earlier sessions. The deoxy triphosphates are formed after deoxy mononucleotides have been synthesized and are transformed to the triphosphate form by group transfer from ATP. So, in the case of deoxynucleoside, the deoxynucleoside monophosphate will undergo group transfer reaction with ATP to form deoxynucleoside diphosphate plus ADP. The deoxynucleoside diphosphate plus another ATP molecule. Both these undergo group transfer reaction to form deoxynucleoside triphosphate with the release of ADP. These triphosphates can then unite to form polynucleotides held together by phosphodiester bond. Okay, so now this nucleoside triphosphate can be utilized for the synthesis of polynucleotide chain. That means they are added to this growing polynucleotide chain of N nucleotides to form a growing polynucleotide chain with N plus 1 nucleotides plus a pyrophosphate molecule. Okay. And this polynucleotides, in the polynucleotides, the nucleotides are connected by phosphodiester linkage. In this group transfer reaction, a pyrophosphate bond is broken and a pyrophosphate group is released. So during this reaction, a pyrophosphate bond is broken and a pyrophosphate group is released. And in this reaction, the delta G is slightly positive. Okay, and during this reaction, the delta G is slightly positive. That is approximately 0.5 kilocalorie per mole. Thus, we know that since it is a positive delta G value, the reaction is in need of free energy. The need of free energy comes from the splitting of high energy pyrophosphate group that is formed simultaneously with the high energy phosphodiester bond. Okay, so and this free energy will be released by the splitting of this high energy pyrophosphate group that is released upon the formation of phosphodiester bond in the case of a polynucleotide chain. This happens with the help of an enzyme which is present in all the cells, that is nothing but the pyrophosphate. So the pyrophosphate get converted to 2 phosphates with the release of free energy -7 kilocalorie per mole with the help of an enzyme pyrophosphate. This large negative delta G means that the reaction is effectively irreversible. That is, once the pyrophosphate is broken down, it never reforms. The union of nucleoside monophosphate groups coupled with the splitting of pyrophosphate groups as an equilibrium constant determined by the combined delta G value of two reactions. That is 0.5 kilocalorie per mole. That is the free energy that are released when the phosphodiester bond in the polynucleotide chain has been formed, plus this -7 kilocalorie per mole. That is nothing but the energy that are released, or the free energy or the delta G value during the splitting of pyrophosphate. Okay, so the resulting value of this delta G will be -6.5 kilocalorie per mole. That tells the nucleic acid almost never break down to reform their nucleoside triphosphate precursor. Okay. Then coming to the importance of this pyrophosphate split. Okay. So the pyrophosphate split characterizes most biosynthetic reactions. The synthesis of nucleic acid is not the only reaction where direction is determined by the release and splitting of pyrophosphate. Okay. So this is not the only reaction. That is, the synthesis of nucleic acid is not the only reaction where this direction is determined by the release and splitting of this pyrophosphate. In fact, essentially all biosynthetic reactions are characterized by one or more steps that releases pyrophosphate groups. For example, the activation of an amino acid by the attachment of AIM, that is adenosine monophosphate, the transfer of high energy bond from adenosine triphosphate to the amino acid adenosine monophosphate complex has a slightly positive delta G. Therefore, it is the release and the splitting of ATP's terminal pyrophosphate group that provide the negative delta G that is necessary to drive this reaction. Okay. The great utility of this pyrophosphate splitting is well demonstrated when we consider the problems that would arise if a cell attempted to synthesize nucleic acid from nucleoside diphosphate rather than from nucleoside triphosphate. Okay, suppose or this the importance of this pyrophosphate splitting is well demonstrated with an example, or if we consider the case of utilization of nucleoside diphosphate instead of nucleoside triphosphate for the nucleic acid synthesis in the case of a cell. If that happens, instead of pyrophosphate, the phosphate would be liberated. Okay. So, if that happens, if nucleoside diphosphate will be utilized for the synthesis of polynucleotide chain, what will be the result? Phosphate will be released. The phosphate will be released instead of pyrophosphate. Okay, this is the normal pathway during which the nucleoside triphosphate will be utilized for this polynucleotide chain synthesis. At that time, by the attachment of nucleotide monophosphate to this growing polypeptide chain, the only nucleotide chain pyrophosphate will be released. But if the nucleoside diphosphate is used, instead of this pyrophosphate, only a single phosphate is released. Okay. The phosphate diester linkages, however, are not stable in the presence of significant quantities of this phosphate. The formed phosphodiester bond will not remain stable in the presence of significant concentration of this phosphate because they are formed without a significant release of free energy. Because these phosphates are not formed as a result of significant release of free energy or associated with release of significant amount of free energy. Thus, the biosynthetic reaction would be easily reversible. So it can be easily reversed. Okay, it can be easily reversed. The reversed arrow mark shown that that can be easily reversed because of the significant concentration of this phosphate. That is, if phosphates were to accumulate, the reaction would begin to move in the direction of nucleic acid breakdown according to the law of mass action. Okay, so if that reaction has happened like this by utilizing this nucleoside diphosphate, the significant concentration of phosphate will be built up, and that will favor the nucleotide breakdown, polynucleotide breakdown instead of synthesis. Okay. Because it is not feasible for the cell to remove the phosphate as soon as they are generated, because the cells require significant internal levels of phosphate to grow. Okay. In contrast, a sequence of reactions that liberate pyrophosphate and then rapidly break it down to two phosphates disconnects the liberation of phosphate from nucleic acid biosynthetic reaction, and thereby preventing the possibility of reversing the biosynthetic reaction. So, in the normal process, the nucleoside triphosphate has been utilized for the synthesis of polynucleotide chains with the release of pyrophosphate. Upon formation of this pyrophosphate, it will be immediately converted to 2-phosphate with the release of higher amount of free energy, free delta G value. Okay, and from that picture, it is clear that the use of this nucleoside triphosphates are precursor of nucleic acid is not a matter of chance. Okay, so upon this, because of the presence of this high amount of free energy that are released as an after effect of splitting of this pyrophosphate into 2-phosphates, this reaction is not reversible. But here, the phosphates are not formed as a result of this release of high amount of free energy. Okay, so from this, it is clear that the use of nucleoside triphosphates are precursors of nucleic acid is not a matter of chance. Okay, so this can be applicable in the case of utilization of ATP instead of ADP as a donor of high energy group in cell. Okay, and this concept is also applicable in the case of utilization of ATP and not with ADP as a donor of high energy group in cell. Before winding up, I wish to correct a mistake. Then we discussed important classes of high energy bonds. There was a small error in that picture of creatine phosphate. See here, this is the creatine phosphate. The creatine phosphate actually contains only a single phosphate. Okay, even though they have shown a pyrophosphate here, but this is the mistake. There is only a single phosphate group attached with this creatine. Okay, so I forgot to correct it when we discussed it earlier. Sorry for the inconvenience.